Amino acids, compare and contrast? I know, sounds like a blast. 20 days of amino acids went by way too fast. So amino acids are protein letters, and they have these different properties that are going to influence how a protein forms and functions. And so by understanding the differences between these amino acids, we can then better understand how proteins work, and I think it's really cool. So let's compare and contrast these guys.
They all have this generic backbone where you have an amino group and a carboxyl group, and then they're going to link up through peptide bonds to form these long chains. And then these chains will fold up to form proteins based on the characteristics of the amino acids, in particular in the characteristic of their unique parts, their side chains or their R groups. And just a little terminology out of the way. Once we link together amino acids, because we're using the amino and the acid parts, we can now refer to what was an amino acid as a residue. So these would be individual residues, although we often use the terms like amino acid and residue interchangeably in the context of a protein. So the different properties of these side chains are going to influence how the protein folds and how it functions. So there's this big connection between the form, so how a protein's like shaped and how it functions. So think of fork versus a knife versus a spoon, and the different properties of the amino acids are kind of going to be helping us like mold the fork or the spoon or the knife or whatever. So it's really important that we understand the properties of these amino acids. Also, acknowledging that the properties of them in their like free form, so when they're just like free floating, are not necessarily exactly the same as when they're linked up. So the things like acidity and basicity, like the pKa values we'll get into, those are going to vary a little based on the context. But one of the, probably one of the most important ways that we can classify amino acids, and one of the things that is most important when it comes to how a protein folds, is the relative hydrophobicity or hydrophilicity of amino acids. Which is going to correlate largely with their polarity. When these atoms link up, they're doing so by like sharing pairs of electrons. And when they share these pairs, they don't always share fairly. This can lead to an imbalance of charge called polarity. So some of the, we call the ones that hog the electrons electronegative, and oxygen and nitrogen are a couple of examples of this. So you'll see that water, it has these oxygens, which are going to be pulling the shared electrons towards it. This is going to make the oxygen partly negative and the hydrogen partly positive. So water is going to be highly polar, and opposite charges attract. And so you can get water forming these big networks, especially because it can form these things called hydrogen bonds, which are basically just a strong partial charge attraction involving an electronegative atom with a lone pair serving as an acceptor, and then a hydrogen attached to an electronegative atom as the donor. And so water can form these intense networks with itself. So it's only going to want to let things into its network that have properties that it likes, things like charge and partial charge. So it likes polar things, it likes charged things. It doesn't like things that aren't polar or aren't charged. So it doesn't like pure hydrocarbons where you just have carbon and hydrogen, which share pretty fairly. And so these are kind of law for the water. It doesn't really offer much. When you have these hydrocarbon chains, they're going to be hydrophobic. And if we have hydrophobic parts of proteins, those parts are going to be excluded by the water. Basically, the water doesn't want to hang out with them, so they're all going to be cinched together in this hydrophobic effect. However, the side chains that have like cool functional groups, so we basically swap out those hydrogens. I think of hydrogens a lot of kind of like space filler, because carbon likes to form 4 bonds, or a double bond is like counting as 2 bonds kind of thing. And so if you replace those hydrogens with something else, something like an oxygen or a nitrogen, which is going to have more, it's going to be more electronegative, so it's going to be pulling those electrons, giving you a partial charge, then you are going to introduce things, you're going to introduce polarity, you're going to introduce things that water will want to hang out with. If water wants to hang out with them, we call them hydrophilic, and we can find these on like the surface of proteins, whereas the hydrophobic things are going to be found in the like in the center of proteins. And so you might think that the hydrophobic things are kind of boring, but really the hydrophobic effect is a large force. It's like the key force in protein driving protein folding, because you can think of as the protein chain is coming out of the ribosome. So as this protein's being made, those hydrophobic parts are going to be like excluded from the water, start clustering together, and this is going to drive the protein folding. Additionally, although that these parts might look really boring because they don't have charge, because the electrons are moving around, like they're just like whizzing around, they can, if they have, you can get basically just like a random, you can randomly have just like a partial charge because the electrons happen to be hanging out in one place versus another. And they can get kind of like a chain reaction where they kind of like all sync up their electrons. And so even hydrophobic things can have interactions with one another that can help strengthen protein. So if we look at the amino acids, we'll see that we have some that are very hydrophobic, such as isoleucine, phenylalanine, leucine, valine. So these have these like pure hydrocarbon chains, so just hydrogens and carbons in their side chains. And so they're going to be nonpolar and they're going to be boring. As we start introducing heteroatoms, so as we start introducing things like oxygens and nitrogens, then we are going to be increasing the polarity. And also, so some of these might seem kind of weird, but you see with like glycine and stuff, it doesn't have all of the carbons and hydrogens. So this is why like you see some of these that are, you still have some of these like pure hydrocarbons up here, but you don't have like the giant hydrocarbon chains. And then once you get into the hydrophilic stuff, that when you have things that are charged, these are going to be very hydrophilic, as well as things with like amide groups. So you have an oxygen and a nitrogen, you have a bunch of polarity, you have things that water likes, you can form hydrogen bonds with, so that's another thing. So it's not just about the polarity, it's also about the ability to form these hydrogen bonds. So when we talk about hydrogen bonding, so with like glutamine and asparagine, they have these amides at their ends that can form these hydrogen bonds. They have these like nitrogens with these hydrogens that can act as donors, and they have those oxygens. So just like, it's kind of like they have the end of a peptide at their back, at their end. So in proteins, the peptide backbone, like how you get those helices and beta sheets and stuff, that's actually coming from the generic part of the protein, from the backbone and its ability to hydrogen bond with itself. So some of the amino acids have the ability to form hydrogen bonds through their side chain. And so this is going to make them more hydrophilic, even if they aren't as like polar as you would think the water would want it. Even if they don't have a charge, if they have that ability, if they have that like partial charge and they have that ability to form these hydrogen bonds, that's going to make water happy. Water will hang out with it, let it into its network, because water's network, remember, is based on hydrogen bonds between water and water. So you can have hydrogen bonds between water and your protein. And so we're going to find those amino acids that can do these hydrogen bonding. We're going to find them in like the hydrophilic. We're not going to find them on the surface of proteins. When we talk about asparagine and glutamine, so they can form these hydrogen bonds. They're really hydrophilic. Water likes to hang out with them, but they're uncharged, and well, they only have like a partial charge polarity. But some of the amino acids are sometimes charged, and water really likes charged things too. Water really likes charged things. If you think a partial charge is good, try a full charge. There are 5 amino acids that we typically think of as being charged a lot of the time. So 3 of these are going to be positively charged and 2 of them negatively charged. But this is all going to be pH dependent, especially in the case of histidine. So these other guys are basically practically always going to be charged, and histidine is kind of a borderline case. So let me explain what I mean. So first we need to address like the backbone charge. So a lot of times you'll see things drawn in this unrealistic form, and I know I'm guilty of it sometimes too, because it's kind of the convention where you draw it, where you have in its non-ionized form. So an ion is something that's charged. So in its non-ionic form, it's uncharged, and that means that you would have this deprotonated your amino group deprotonated and thus neutral, and your carboxylic acid group protonated and thus neutral. But there's a problem with this. This is like not realistic at all. Instead, how you're normally going to find it is in this zwitterionic form. So we call something a zwitterion when it has like a positive charge and a negative charge, but they cancel out. And so you have a neutral molecule overall. So don't confuse this with polarity. So this is polar, but with polarity, we can also just have like partial charge separation, whereas with the zwitterion, you have full charge separation, but neutral overall. The basic idea is that whether these are protonated or not is going to depend on how many protons are around, which is measured by the pH. So the amino group is going to want a proton much more than the carboxylic group is going to want a proton. And so if there are enough protons available to protonate this carboxylic group, this amino group would already have taken one a long time ago. And if there's not enough protons for this amino group to have one, well, this carboxylic group sure ain't going to have one. And so you're almost always going to see this zwitterion form in our bodies. And it's unrealistic to think of amino acids as being in this non-ionic form, although this is typically the convention of how they're drawn. And so the reason why you have this is because this amino group is a lot more basic than this carboxylic group. So an acid is something that takes a proton, and so an acid is something that donates a proton, and a base is something that takes a proton. Whether something takes a proton is going to depend on how much they want the proton. So things about the chemical nature of the group, as well as how many protons are around. And so how many protons are around is measured by the pH. So the pH is the measure of how many protons are around, and it's this negative log, this inverse log scale. So basically, the more protons you have, the more acidic the solution, the lower the pH. The fewer protons you have, the more basic or alkaline the solution, and the higher the pH. And for every one change in pH of 1, you get a 10-fold change in the proton availability. So if you go from like about 7, like our bodies, to an 8, you'd have 10-fold more, 10-fold fewer protons. And if you go to a 9, you'd have 100-fold, 10, you'd have 1,000-fold, et cetera, et cetera. So then that part is, the pH is talking about like the solution level. But then different groups are going to have different willingnesses to take a proton. And so some of them will take a proton even if there's not very many protons available. And we call these guys basic. Some of them will take protons if there aren't very, won't take a proton until there's like a ton available and they really can't escape them. And so these are typically referred to as like acidic. But it's important to note that basically once you give up a proton, now you're the conjugate base, you can act as a base. Once you're the conjugate base and you take a proton, now you can give a proton, so you're the conjugate acid. And so we can refer to both forms of a conjugate acid and conjugate base as like being flip sides of the same coin and the same molecule. And whether one wants to be on one side or another is determined by, like, we can measure that this value called like the pKa, which is going to tell you at what pH do you have an equal concentration of each of these. So if you're above the pK, so a strong acid is going to have a low pKa, and this means that even so you have to get it to really, really low pH in order to protonate it. So it's almost always going to be in its deprotonated form. Whereas something that's more basic, stronger base, you're going to have to get it to a really, really high pH, so where there's really not very many protons at all available before it'll give it up. So if we look at an amino acid, this amino group, it's going to have a pKa of around 10. So we'd have to get to a really high pH in order to deprotonate it. And so if we're at a pH of like 7 in our bodies and it has a pKa of around 10, so we're about 3 below its pKa, we're going to have like 1,000-fold more of the protonated to the deprotonated form. And when that amino group is in its protonated form, it's positively charged. Now let's talk about the carboxylic group. The carboxylic acid has a pKa of around 2. So this means that at a pH of 7, we're at like 5 above. So we're at like 100,000 times more. It's going to be in the deprotonated form. So you can imagine like doing things to change the pH. But what's going to happen is that if you raise the pH, so if you raise the pH, you have fewer protons available. Sure, you're going to deprotonate this amino group, but this carboxylic acid had deprotonated a long time ago. And if you go to a lower pH, you go to a really low pH where you have lots of protons available. Yeah, this carboxylic group will take a proton, but this amino group had taken one a really, really long time ago. So the basic thing is that if you go to a low pH, you're going to have a positive charge overall. If you go to a high pH, you're going to have a negative charge overall. But normally you're going to have this zwitterionic form where you have plus and minus charges that cancel out, and you're like never going to have this non-ionic form. So our backbone has charges, but only at those ends, remember, because when they're linking up, they're doing it through this. So when we talk about free amino acids, free floating, then they'll have these charges, but when you have it in a protein form, then you're just going to have these carboxyl amino groups at the ends. So it's still charged, but you only have like one on each end. So we call the N terminus the one that has the amino group, the nitrogen, and then the C terminus is the one that has this carboxyl / carboxylic acid group, which, as we just talked about, is typically going to be in this carboxyl form. But these charges cancel out. But some amino acids have charges in their side chains. So they have a third pKa of their side chains. So basically there's 3 that we call basic, and these would be the positively charged ones, the ones that can be positively charged. And then there's 2 that we call acidic, and these are the ones that we're going to be found negatively charged. And so the reason why we call these like basic versus acidic is because in one definition, an acid is something that donates a proton, and a base is something that accepts a proton. So if you have an acid, it donates a proton. If the acid was neutral, now you have a negative charge. So we refer to aspartic acid and glutamic acid as acidic, and then once but they typically find them after they've already like acted as an acid. So we find their conjugate base, aspartate and glutamate. With the basic ones, basic refers to their ability to take a proton in their neutral form. So in their neutral form, they take a proton. Once they take a proton, because the proton is positively charged, now they become positively charged. And this is the conjugate acid. So now they can like act as an acid. But you see that they can go back and forth between their acid and their conjugate base. And this is going to be dependent on the pH, which is a measure of how many free protons are around. So if we look at these amino acids with the quote unquote acidic ones, we have pKa's of around 4. In our body, we're around 7.4. So basically, we're several orders of magnitude above their pKa. And if we're above their pKa, we're going to be in the predominant, like almost exclusively, if we're that far above the pKa, we're going to be almost exclusively in the deprotonated form. So you'll usually find these in like the aspartate and the glutamate forms. When it comes to the basic amino acids, so with lysine and arginine, these guys have really high pKa's. So we're always going to be far below their pKa. And so if we're far below their pKa, this means that we're going to be in the protonated form, and for these, their protonated form is positively charged. When it comes to histidine, so histidine's pKa is closer to the bodily pKa. And so we have about a 1:10 ratio of the deprotonated neutral form to the protonated form, positively charged form. But it can go back and forth, and it plays important roles in like enzymes in their like active site, giving and taking protons to help make reactions happen.
Really key thing to remember is that the basic versus acidic, in terms of how we classify them, is based on their neutral form. So in their neutral form, the basic ones act as a base and take a proton. So when you see them protonated, they've already acted as a base, and now they're the conjugate acid and can act as an acid. Contrastingly, if the acidic amino acids, acidic refers to their neutral form. So in their neutral, their carboxylic acid form, they were able to act as an acid. So they acted as an acid. They deprotonated. Now they're negatively charged and they're the conjugate base. So they can act as a base. So remember that the naming is referring to the neutral form.
So there's also ones that we can deprotonate. It's just a lot harder. So with serine and cysteine are a couple. We'll compare these because they are basically the same, except this one has an oxygen, this one has a sulfur. So it's going to be a lot harder to deprotonate this OH group than it was when we had like a carboxylic acid group. So with the carboxylic acid group, when you deprotonate it, you have resonance, so you have sharing of this charge. With the hydroxyl group, you don't. So this hydroxyl group, this oxygen, you're going to have, it's because oxygen, so like an electronegative and stuff, you're going to have this high concentration of charge. This is going to be very basic, so it's going to want to take a proton. And so this is really reactive, but it's also harder to form because it's less acidic. It's not going to want to give this up. But in the active site of enzymes and stuff, you can get this deprotonated in order to then like serve as a nucleophile. So something that has more electrons in it once and then can attack an electrophile, and then you can add things and that sort of thing. Much more on that in other posts. With the cysteine, you have a sulfhydryl or a thiol group, so you have a sulfur. This is going to be easier to deprotonate. So has this like bigger, it has this bigger electron cloud. Basically, it can better spread out its charge, and so this is going to be, it's going to be shielded less by caging effects from the water, and so this is going to act as a strong nucleophile, more nucleophilic than this O-, and it's also going to be easier to form because this bond to the hydrogen is weaker. So much more of that in the post on sulfur. So whereas a serine, it's like super duper hard to deprotonate, pKa of around 13. With the cysteine, we have a pKa of around 8.3. So it's definitely easier to deprotonate this. And then when we see like we have the like this weirdo called cysteine, that can sneak in, it has an even lower pKa. Now let's address the elephants in the room. Some of these amino acids are huge, like tryptophan, this guy's giant, whereas glycine, it just has a hydrogen. So there's a wide range of sizes in the side chains of amino acids. And this has important consequences because the angles that the protein backbone can take are largely going to be determined by the size of those side chains. So amino acids, the peptide backbone of proteins, is highly restricted because these peptide bonds holding them together have this partial double bond character. And in order to have that character, they have to be like in a plane, and you can't rotate around a double bond. So you can't rotate around this partial double bond character. You can't rotate around the peptide bond. You can only rotate on either side. But how much you rotate is going to be influenced by the size and properties of the side chain. So if you have something small like glycine, you can go really flexible. Whereas if you have something big and bulky like tryptophan, you can't rotate as much. And so there are characteristic angles that different amino acids like to take, and this helps influence what type of secondary structure they can be in, like helices and strands and that sort of thing. So two that are the most like different than the others are glycine, which has just this hydrogen and it's really flexible. And then proline, which has its side chain is going to like loop back on itself, and so it can like stabilize regions. It has this high restrictedness, and you often see glycine and proline kind of like acting together to help like form like kinks and that sort of thing. Another thing about glycine is it's the only one that's achiral, and so basically chirality is when you have, so you get a chiral center where you have a carbon attached to 4 different things, and you can think of them as being attached kind of like sticking out in different directions. And so there are different like stereoisomers, which is where you have like different versions, like different handedness. So kind of like your right versus your left hand. So basically almost all the amino, our bodies use like the L form of amino acids, but for glycine, it only has like a single stereoisomer because it's alpha carbon, so the central carbon that's attached to things. It like attached to the side chain, attached to the nitrogen, attached to the carbonyl carbon. So in most of them, it's attached to 4 different things. With glycine, you're only attached to, you have 2 hydrogens, so it's achiral. So glycine is the only one that is achiral. Tryptophan isn't just huge, it's also aromatic. And so what we mean by this is that it has, its electrons are kind of like delocalized around this ring. And so much more on that in other posts. But you get this resonance stabilization where you kind of have the electron shared like above and below the planes. This is going to give it properties like making it like absorb UV light, which is really helpful for like detecting it in the lab, detecting protein concentrations and stuff in the lab. And it's also going to offer it binding opportunities with things. So some of these like leucine and isoleucine, these guys are exactly the same size. They just have their different rearrangements basically of their side chain. So they're the same size molecular weight wise, but with isoleucine, you have this branching closer to the backbone. So it's going to be more restricted in its motion. So these are 2 of the branched chain amino acids. The other one we talk about is valine. And so these guys all have to go through like special breakdown pathways because this is harder to break down in addition to being sterically hindering. So basically steric hindrance is basically where you can't have 2 atoms occupying the same space at the same time. So it's kind of like the atomic version of, hey, I was here first, give me some elbow room. Speaking of breaking things down, we can classify amino acids based on what their breakdown products can be used to do. So broadly speaking, we can refer to things as glucogenic or ketogenic or both. Although in reality, these pathways are all like really, really interconnected. So you can typically always make whatever you want to make in some form, but it might involve like using different parts of things. And so you might not have like a net gain of the thing. So then we don't call it one thing or another. It's kind of complicated. But basically when we refer to something glucogenic, that means that we can break it down to something that can be used to, something that we can break it down to a component of the tricarboxylic acid cycle, sometimes called the TCA cycle, Krebs cycle, citric acid cycle. And this is the cycle that it can be used when you're breaking down glucose, but it can also be used to make oxaloacetate, which can be used to make glucose. So if something makes something that goes in that's in the TCA, then we call it glucogenic. Some of these make, when we say something is ketogenic, it can be made for making lipids like fatty acids, as well as making these things called ketone bodies. So ketone is where you have a carbonyl, so C double bond O, with 2 carbon groups on either side. This one's not technically a ketone, but we still call it a ketone body. But anyway, so this is like an alternative energy source, more on that in other posts. But basically, so some of these amino acids can be made into those type of products that should not be used to make lipids. There are 2 that we refer to as like exclusively ketogenic, which are leucine and lysine. And then some of them are both ketogenic and glucogenic. Another way we can classify amino acids is based on like what we can make, if we can make them or not. So you might have heard of like essential amino acids. So basically that's not referring to whether or not we need them. We need all of these. But it refers to like essential in the dietary sense, like whether our bodies can make them ourselves or whether we need to get them directly from our diet. And so there are some that are essential that we need to basically get pre-made from our diet, and then others we're able to make from other amino acids or from other products of metabolism, so like the making and breaking of molecules. we've been talking about amino acids in like the form that they're used in translation. So like the form that they're put into the proteins as, but some of them can also get modified after the fact. So we call these post-translational modifications when they occur after they've been added to the growing protein chain. And this can be used for like regulation and that sort of thing. And so some of these amino acids are going to be able to be modified in different ways. So one of the most common post-translational modifications is phosphorylation. So this addition of this phosphor group, which is negatively charged and bulky, so this kind of can influence how a protein folds, like whether it has to like change shape a little to accommodate it. It can create binding sites for various things, a lot of cool stuff. And this can happen on like typically there's three main ones: tyrosine, serine, and threonine. So histidine can also get phosphorylated in some rare cases, but it's hard to study. And then lysine can get methylated, acetylated, which is really important for the like epigenetics. So basically making changes to the proteins that DNA is wrapped around to help influence whether they're accessible or not for transcription factors to make messenger RNA copies of them to make proteins from. And finally, I need to mention a couple that aren't on that initial chart. So we have 20 like canonical amino acids like that are genetically encoded, so they can be used to, they go into proteins. So amino acids, there can be amino acids that are non-proteogenic. So basically they technically amino acids because they have like amino groups and carboxylic acid groups, but they don't serve as in proteins. And we also have like derivatives of amino acids that do serve in proteins that can be used in other functions. But when we're talking about like the genetically encoded ones that we put into proteins, we have those 20 main ones. We also sometimes have incorporated selenocysteine. So basically it's like cysteine, but it has the selenium instead of a sulfur. So this is only in a few proteins in our body, and it kind of has to sneak in. It doesn't have its own codon. It uses a stop codon. So more on what this means in a second. And then pyrolysine. So this guy's just found in like some archaea and bacteria that break down products into methane. So these methanogens. And so we don't have this, but they do, and they do by using a stop codon as well. But they have different strategies. So basically in translation, the ribosome is going to go along until it sees a stop codon. And so a codon is where you have like a 3-letter mRNA word that's spelling the amino acid. There's a few that don't spell an amino acid and they instead tell the ribosome to stop. What happens is that in the case of when you want to put a selenocysteine in, the RNA, the messenger RNA, it has this special element and that's going to bind to these like helper proteins and bind to this tRNA that's loaded with the selenocysteine and kind of sneak it in at the stop codon. In these archaea, basically they don't use this as a stop codon anymore. They basically just use it for this pyrolysine. So then they don't need all this special machinery in order to sneak it in. What's really cool is that scientists have found that you can basically take over the ability, this machinery that's used to put in this pyrolysine and use it to add like unnatural amino acids to do really cool stuff. And so I think that's a really exciting avenue of research, because although the amino acids that we have are super duper awesome and we can do a lot of cool things with them, as we've seen, we can even expand the toolbox with these unnatural things to do various experiments in the lab. So remember, know your amino acids.